Hello there, what do we have here? We have a nice little diagram showing what happens during the first stage of protein synthesis, which is what? Transcription. What happens during transcription? Transcription, we think copy. Something's being copied. What's being copied? One DNA gene. What form does the copy take? One messenger RNA molecule. So what happens during transcription? A copy is made of one DNA gene forming one messenger RNA molecule. So that messenger RNA molecule has a message. What is that message? The information in what? One DNA gene. Yes, indeed. Okay, so enzymes open up a DNA molecule at the beginning of a gene. And that looks a lot like DNA replication, but it's a very totally different process. But it looks a little bit similar right now. So the enzymes somehow know where the genes begin. How do they know that? Well, they've been to enzyme school. No, we'll get a little bit of an idea of that in a little farther through this unit of how they know where the genes begin. But the enzymes open up the DNA at the beginning of a gene and start transcribing the gene. How does that work? Well, that works like this. RNA nucleotides are attached to one side of the DNA molecule. Gee, only one side is transcribed. How do the enzymes know that? Well, somehow they just know. And so this is a little different than DNA replication because we're dealing with RNA, which is not at the golf course, but at the university golf course, right? And so here, everything is the same, A's with T's with G's with C's, except for RNA, there'll be a U instead of a what? A T. And so we can see that. We see here a C attached to a G to a C, but a U to an A, not a T, but a U. Another U to an A, and then C to G, and A to T. T's, A still connected, DNA T's. And so it goes on that way. You do it exactly, we'll do practice in another pencast, but it's always just like DNA replication, except U's instead of T's on the RNA side. And so on it goes. This is called the replication bubble. This is not the replication, but the transcription bubble. And on it goes, and we have this RNA right here. This RNA right here, what's it a copy of? Well, it's a copy of the gene that's already been closed back up. And so on and on we go. More and more transcription. How far does this go? Well, what's being copied? One DNA gene. So how far down does transcription go? Until what? Until the end of the gene is reached. Yes. How do the enzymes know when the end of the gene is reached? We'll find out. We'll find out not in this video, but soon. And so what do we have down here? We have an RNA molecule, a messenger RNA molecule. It's a copy of a DNA gene. The DNA is being closed back up. What's the next stop for Mr. RNA? On out of the nucleus into the cytosol to get grabbed by what? Our ribosome. So that, ladies and gentlemen, is transcription. Let me ask you this question. Where is the RNA molecule before transcription? I asked this in the class, and I get lots of puzzled looks, and occasionally a confused student. But it's a little bit of a tricky question, so that's okay. So where's RNA before transcription? Well, I see RNA down here. Do I see any RNA in this first picture? Any messenger RNA? No, I don't. And why not? Because it's this process that creates it. This process of transcription creates the messenger RNA molecule. So where is messenger RNA before transcription? It's nowhere. It's this process that creates it. All right. That's it on transcription.